Good evening to everyone. Now it is almost 9:45 time, and today is actually the date is 28 May 1921. Today we are going to learn an important concept called heredity. Then we also try to understand genetics, especially now there are two types of genetics we have to discuss. One is the classical genetics, second is the modern genetics. We shall examine what they are very briefly. Coming to heredity, it is just passing on the characters or traits from one parent to from parents to offspring. That is heredity. We know that sometimes people say, my nose resembles my grandfather's nose. I might not have seen my grandfather, but people who have seen would say like that. So like that, in almost all living beings, which are multicellular and what we call eukaryotes, and also prokaryote single-celled animals, there is the process of passing on the parental characters to offspring, but in different ways. Study of that, how the characters are passed on from one generation to another generation, was not known earlier, and the study of that particular thing is called genetics. Genetics started much earlier. We'll look into that one. So, scientific explanation of how traits are passed is genetics. Now we can think there are two types of genetics, and one genetics are what we call the classical genetics. Here, what we call the classical genetics, it almost started almost in 1760, and on the timeline continued up to 1860. This is the timeline when classical genetics was operating. That means people were studying hybrids, people were studying pea plants, people were also studying some animals like moths, maybe honeybee, and all these things. That means there was an attempt by the earlier scientists about to know how genetics happens in these things, but it was not very clear because at that time there was no information on either chromosomes or on DNA or on genes. However, in the around certain time, like 1856 onwards, 1822 to 1884. This is the period when a Austrian monk called John Gregor Mendel, studied systematically pea plants and also honeybee. However, he was not successful in studying honeybee, but he was successful studying the pea plants. Pea plants are bisexual. They have the capacity to self-pollinate and also cross-pollinate, and their lifespan is very short. Within say 1 year, their lifespan is 1 year. In 1 year, they can produce seeds, and seeds can grow. So pea seeds have 7 very clearly observable traits or characters. So there are 7 observable characters in this one. So John Gregor Mendel studied this almost for 10 years. That means he must have studied the pea plants reproducing and producing plants, again reproducing, producing again and again, like that. He must have seen more than 30 to 40,000 generations of the pea plants. That means while doing all these things, he came out with a model, actually, what is normally called the study model. Mendel came out with a scientific model called the study model, because he was able to study more than 30 to 40,000 pea plants, and he was a mathematician and also a biologist. So this helped him to quantify using ratios about the genetics. So now people call Mendel as the father of the modern genetics. He is called the father of modern genetics. Father of modern genetics. Modern genetics. This we must remember. Now, Mendel's work was mostly on the appearance of the plants. That means how tall, how short, what is the color of the flower, where is the flower situated, what is the color of the pod or the fruit of the pea plant, whether seeds are wrinkled or smooth, like that. 7 important characters he studied, seven traits as they're called, or 7 important characteristics he studied. So we had, for these things, there were opposite plants also. If there was one tall plant, there was also a short plant. So there were tall as well as short plants. Similarly, there were plants producing pink flowers and plants producing white flowers. There were some plants producing pod will be yellow, some will be green. Like that, the 7 opposing traits were there. It becomes very handy for him to study, and by studying all these things, he came out with what's called the Mendel's activity was completely based on these things, and from these things, Mendel came out with what is called the principles of inheritance. So this was possible for him because he was a mathematician. He systematically made a model of his own study and he followed that. That model included taking the pure plants. I will tell you what is the meaning of the pure plants. He was taking pure plants, then he was crossing them. Then he used to get the generation are called the offsprings, and these offsprings were maybe many generations he was getting, generations of plants he was getting, and then he used to self-pollinate them, and then again get many plants. So that means he was trying to repeat whatever he did in one plant, and then he got the data. So his work is very, very important, and he took patience and he did a lot of work on the pea plants. because pea plants are very, very easy to handle. Number one, they have a very short span of life, and they contain 14 chromosomes. Now we call it chromosomes. Earlier he called it the triads, or he called it the elements. He called it elements, or you may even call it units. At that time even the number was not known, but there were definitely 7 triads were known. 7 triads were known at that time. Now that is that type of work where appearance was used to study how inheritance takes place without having known anything about the genes, or talking only about the units or the elements. And that kind of genetics is called classical genetics. It lasted almost for almost long time. After 1884 onwards, when by this time people started finding out more and more about the cells, and more about what is inside the cell. So by the time around 1884 or 85 or 86, they discovered the nucleus. They called nucleus scientists, and they found out inside the nucleus there is what's called the chromatin, and they also found out chromatin becomes into pieces during the cell division, or what you call during the reproduction of the cell, and they called it the thread-like structures. And later on, around 1870 or so, 75, name chromosome was given. That means there was a good progress going on the especially to find out what is in the nucleus. At the same time, Watson and Crick did a lot of work, and they explained that the material inside the nucleus is DNA, and that DNA looks like a double helix. So this information all came when these things are all going on, and the 19th century, 20th century, and the 21st century. Today we have what's called the modern genetics. Modern genetics means all the observations what we do is based upon the gene. Gene again is present on the chromosomes. We will come to that at a later stage. So now the gene is the basis for heredity, and this has opened up a lot of studies. We have today humans can be studied, rats can be studied, plants can be studied, microbes can be studied, animals can be studied using genes. So we today we have in the modern in the modern genetics, we can study gene expression, gene interaction, sex determination can be taught using the genes, linkage of traits, sex linkage, crossing over of chromatids, cytoplasmic inheritance, polygenic inheritance, genetic engineering or recombinant DNA, cloning can be done, gene therapy is possible, genetical modification is possible, and recombination of viruses are taking place. All these can be studied in different parts, especially using gene as the basis for the genetics. Therefore, modern genetics has a large amount of openness to do research and find out more and more. Though it is there, still the Mendelian ideas are used, and people would certainly like to use the Mendelian ideas. Now, Mendel, when he gave three important ideas, that's what I think you should be able to remember. Mendel called them principles of inheritance. That's what he called. But later on, due to lot of research, repeated research, and every time it became true, so therefore it became the laws of heredity. So he proposed three very important ideas, which we call them the laws of heredity today, or the Mendelian laws. One of them is called the law of dominance. This you must remember. Second is called the law of segregation. We shall know what they are very in the next few moments. And the third one is law of independent assortment. So these were the three important propositions he made, which later on became the laws. Even today, in genetics, these three laws are applied, but in organisms like human beings and higher animals, since the number of genes are more, for example, in human beings, there are 46 chromosomes in man, or by birth, that matter maybe women also. 46 chromosomes are there. On each chromosome, there will be 30,000 genes. You can imagine how many genes are there. And one set of chromosome, that is 23 chromosomes. 23 chromosomes is called the haploid set, and whatever the number of genes are present, it's called the genome. So there are two genomes in the human beings today, and therefore it is very difficult to use these three laws of Mendel. But to some extent, we can use them in even now in the organisms like Drosophila, or some organisms where number of chromosomes are less. For example, in Drosophila, only 4 chromosomes are there, where the number is 2n or 2x number. Now let us try to understand what these three are. Law of dominance, law of segregation, law of independent assortment. I will not take much time, because you can study these things in your own textbooks are given there. If textbooks are not available, I think the best source would be internet. If you go to internet, you will be able to do a lot of work. Now let us see what Mendel did. So Mendel was doing what's called breeding experiments. Breeding. What does it mean? That means he used to cultivate the plants, especially pea plants. Pea plants he took, and he was trying to use his own intelligence to pollinate them. Now, what is that he did was, he took very, very simple traits. He experimented all the 7 traits, but let us take the example of one trait. Then let's take the example of height of the pea plant. When you take up the height of the pea plant, you may find a tall pea plant. Many plants are tall plants. I'm using an arrow here. Another one may be smaller than that, and that's also a pea plant. And so one is tall. This is short. We can say short or dwarf. So pea plants he selected. So what he did was, he selected in such a way that he observed many plants, pea plants, and he selected plants, seeds. When these seeds were sown, it only produced tall plant. Let us use the word symbol T for tall now. Okay? So it's only tall plant. So he called such plants as pure plants or breeding true one trait. So these seeds always produce, wherever you put them, they always will produce the tall plants. And he took this generation of plants and he collected the seeds. When you put these seeds in the soil, again these produce only short plants. So these were called pure type. So he selected, see his model study is very correct. He selected, whenever he did experiment, he was using the pure plants only. Now let us say, use the word now in a different way, same thing. Let's say the parents, he took the parental plant, we say P as the parent. Parent was tall. Now we are writing the phenotype. That means I am using another word called the phenotype. Phenotype refers to the trait, what we could see by simple observation. If you could see my skin is dark, my phenotype is dark. If you could see an American and his phenotype is white. So observable characters, when you describe, we call them phenotypes. What cannot be seen but still it is important, that will come to that later on. So he took the phenotype tall and also phenotype short. The plants he took, they are the parents pure. Now then he made a cross-pollination. He used cross-pollination. I think by this time you know cross-pollination refers to the transfer of pollen of one plant to the other plant. That's called cross-pollination. If the pollination happens in the same flower or in the same plant, it is called self-pollination. So first time he made the cross-pollination, then he collected seeds. Now when he collected the seeds, he planted them and he tried, he got all plants he got were tall. Now look at this. All plants tall. He called this as the F1 generation. F1 plants. F1 or F refers to filial, means sun, like sun, you can call it. F1 plants. All plants were tall. Then the question came to him, what happened to this shortness? Where it has gone. So he, in order to explain those things, then he said that must be hidden. That must be hidden, or it is recessive. Then what he did was, he took the F1 plants and then he made self-pollination on this one. That means use the plants, same plants to self-pollinate them, and he collected the seeds again. Seeds were collected, he put them in the plant thing, then he got the F2 generation. That is second generation. Progeny or second generation progeny came. This is the parent. From parent we got the first one, then we got the second one. And this, in the F2, he analyzed mathematically, he got a great number of plants. He got 3 tall plants and 1 short plant. So now what happened is, the trait shortness that was hidden in the F1 generation now shows up in the F2 generation in a very small way. So this type of study he made, and in this he come to a conclusion, if we study one trait then one will be dominant. It expresses well, other will be recessive. That will not be shown. Thus, the law of dominance came into being. Since he did not know about the chromosomes, he felt that these seeds must have in their germ cells, let's say in the germ cells or the pollen grain, must have the factors for tallness. He said there must be two factors for tallness, that is maybe in the germ cells, or the cells that will produce the pollen grains or the female gamete. And he also felt that the recessiveness may be having the smaller t. So these two factors he identified. So he said that in this particular case, there may be one tall factor and another one short one. What there? So like that. So when he again put them into the all plants, again when he tried, he imagined he got 4 plants. Let me say 4 plants. Why I am telling 4 plants? Because all tall. Because when the pollination takes place, when the fertilization takes place, when they divide into four fertilized portions will come. These 4 will become plants. So this was the study made during the meiosis. You can study meiosis, you understand this much better. So according to Mendel, when pure tall and pure short were crossed, he got the F1 generation. And this generation, for appearance, the phenotype, they will be tall, but their internal part, the factors, what they call, is called genotype. So at that time he used the word factors, still it is genotype. What is present inside the cell is the genotype. So genotype will be like this. When the genotype has one from one parent, another one from another parent, and this is called the hybrid. So this is hybrid, whereas this has 2 factors. We said this is the pure one and this is also pure one. So when 2 pure are crossed, we get hybrids. When hybrids are self-pollinated, then we get 3 tall. When these 3 tall were examined genotypically, he found 1 like that, 1 like that, another one like that, another one like that. So the 4 plants, if you study genotypically, 1 is pure tall, 1 is hybrid, third one is hybrid, 4th one is again pure short. So we get 1:2:1 ratio, that is genotypic ratio. So he found out that the crossing and by self-pollination, he was able to identify that the triads in the pea plant would be definitely producing either tallness or shortness. This is what the first law of dominance tells. Similarly, he studied later on for 2 triads. After that, I will rub it off so that we will just say a few words about the next 2 ones. Okay? So now he came to, he took 2 contrasting triads for his experiment. He took, for example, say a tall plant with and and say a red flower. He took another short plant and maybe white flower. White flower. So now if you write for this one, this is the pure plant. It is the P. That is pure parent. I told you already he has developed a model of study. In that model of study, the first one will be always the pure plants. So if you write the genotype for this, it will have two factors for the color, red color. That is the genotype. For this, two small t's and white can be shown as small r. So that is the genotype. This is the phenotype. So when he did cross these things and then tried for F1 and then F2 generations, he ultimately got 9:3:3:1 ratio. This is the ratio he got when he analyzed the F2 plants. So that means 9 plants were tall and red, 3 plants were having tall and red, 3 of them showed short and white, and one was completely short and white. So this is called dihybrid ratio. He did. So by doing all these experiments, he found out that the tallness and the red trait may not go together. They independently sort out and segregate. So by these experiments, he came out with the law of independent assortment and law of segregation. So that is the work of John Birger Mendel, which perhaps you will be able to study from your textbook, and then we will just wind it up by telling that even today that almost in almost all studies of the plant and animal, this is applied. When we come to the modern genetics, we should know that a nucleus contains, imagine that is a nucleus in a cell. Nucleus may contain inside the nucleoplasm, and inside that there will be a long chromatin. This chromatin at the time of cell division, they divide into small thread-like structures like that. These small thread-like structures, which are all dividing into chromatin, will be chromatids. Chromosomes. So each chromosome will have a central centromere attached to that one. Each will have 2 chromatids. 1, 2. They are chromatids. Now these chromatids, when you examine them, they will contain a long chain of DNA molecules. Very long chain of DNA molecule, which may be very, very long one. And these long chain of DNA molecule will, if you take one of them, they will show here like that. If I write like that, they will show 1, 2, 3, 4 like that. 1, 2, 3, 4, 5, 6, 7, 8 pairs per turn of what they call base pairs. So DNA contains base pairs. DNA also contains the sugar. It also contains a phosphate. So today we know that the DNA is made up of phosphate, sugar and base pairs, or we can simply call them, they are made up of nucleotides. Nucleotides. That is, if I write like this may be a phosphate. To this, you will have a pentose sugar. To this will be connected a nitrogenous base. This is a nucleotide. So like that, you will have large number of nucleotides pairing each other, nucleotides pairing each other, and that is called the base pairs. There will be very number of base pairs in that, and some length may be a shorter form. Supposing if you take this chain from here to here, maybe this may be one segment. This segment having a number of nucleotide sequences will be called by name gene. So gene is a segment of DNA, and this gene may be responsible for many things. Now I told you that the pea plants will always become tall and some plants always become short, we said. How it is possible? If you use the gene information, now we say gene will induce enzymes in an organism in this plant, and these enzymes will produce proteins, and these proteins will be responsible for the height of the plant. That means we can say today that the gene expression or gene control is responsible for tallness. Like that, the study of the genetics using the gene as the basis is known as modern genetics, that you will be studying in your higher classes in much more way than what I taught to you. Thank you. Please read your book, try to understand more about it.